When I was a young botanist, in some of my first botany jobs, growing plants for the Forest Service doing restoration, one of the things I kind of noticed about a lot of the plants that would come up and start to bloom, especially a lot of the members of the sunflower family, would get these very bizarre kind of elongated flowers that we call fasciculations. And what I really realized soon, just kind of doing some kind of thinking on my own, was that a lot of these came up in plants that we collected from our gardens. So ones that were heavily inbred likely. And we see a lot of these mutations like this through a lot of different plant groups. And the exciting thing about these mutations is a lot of them aren't going to go anywhere. They're just kind of weird anomalies on the side of the road that we pass by. But when it comes down to it, though, a lot of what we see today are the results of mutations from other things, but they're mutations, unlike these, that have very important consequences for the evolution of that plant. So we're going to start talking about the angiosperms this week, which are an amazing product of evolution and really one of our greatest and most diverse groups of plants that we have. But before we jump into that and start to really get into what an angiosperm is, I think it's important to take a step back and think about all the things that had to happen and for us to get to this point. So we have to remember that plants themselves are products of evolution. They've been shaped and since they came onto land way back here down with the green alga. And plants will inherit different characters or traits. And that these traits are what we consider character states. So you can have a lot of different kind of states within the same character, and so variation within that trait. Along with these, we have adaptations. So these are phenotypes, so expression of certain genes or traits that help an organism deal with various biological or abiotic challenges. So for instance, we've talked a lot about these adaptations in the context of how are these essentially aquatic plants able to now live on land. And this idea of evolution and adaptations and traits happen at the population level. So a group of inbreeding organisms and the variation within that group is where we see these changes and adaptations come across. And of course, evolution itself is just a change in heritable characteristics. So they need to be characteristics that can go from one generation to the next and are going to be acted on in successive biological populations. So a lot of what we talked about, you might ask, how do we know this? And a lot of those ideas of what we think plants were like back maybe in the Silurian period come from Paleobotany, or the study of these ancient fossils, such as this petrified lycopod from Petrified Forest National Park. But of course, the fossil record is very incomplete. In fact, a lot of what we see as fossils is probably likely not a good representation of what was going on, because a certain process had to take over to fossilize and preserve those material. And so a lot of what we can understand from evolution comes from kind of these indirect sources, especially things that we call homologs. So we look at extant organisms and look to see what traits they share in common. And we use these kind of common traits in order to get an idea of the sequence that might have led to these extant animals or plants, so we can make these conjectures. We also looked at shared metabolic pathways. So for instance, if a plant does C4, it's likely that its nearest ancestor might have evolved C4 too, especially if it shares a sister species that does that as well. And also we look for similarity in different DNA, especially plants that have these nice three genomes, whether they be the chloroplast, the mitochondrial, or the nuclear genome. But we have all these ways to kind of look at these relationships, but it's really important to think about how these relationships form. And when we think about plant evolution, we need to think about these various alleles they have, which are a particular form of different genes. So if we take these two theoretical plants here, they have different alleles for flower color. They have the same shape, same basic layout, but the only thing different is this one trait with colors, which could be on the same gene. And so we have these two alleles. We would also call these different genotypes because these two individuals here have different genes and different alleles on those genes, and so that they can be these different types. Along with this genotype is the phenotype, which is the outward appearance of that gene. So in this case, the outward appearance is, again, the flower color here. And these kind of different features and different ways to look at this given genetic information and expression can also be used by looking at what we call their fitness. And fitness isn't about how much these guys can bench in the gym. It's about whether or not they're able to survive and reproduce. So it's more of an evolutionary fitness that we're talking about here. So let's think about how evolution might work on this population. So we'll take a group of these plants here, these theoretical plants, and we have a bunch of different phenotypes. We have a couple yellow-flowered ones, but most of this population is these orange ones. And if we think about this population, let's think about what their common ancestor might have looked like. We don't really know what the flower color was, but they have a lot of similarities, or those homologs in shape with each other, maybe leaf size. But here we'll demonstrate this with flower shape is the same. So that's considered most likely a homolog that unites this population. But what we're going to do with this population is we're going to start to mess with the allele frequencies. So the proportion of essentially these orange flowers to these yellow flowers. Because in this population, as we change these allele frequencies, we're actually going to make this population evolve into a different maybe species, or at least just change their allele frequencies. So the first way that we have evolution works is through mutations. And this is just a change in allele. Maybe mitosis or meiosis got messed up. Maybe there was some kind of outside influence that kind of tweaked or mutated a gene. There could be a lot of ways that these mutations happen. Most of the time, these mutations are fairly neutral. They don't have any impact on the gene. And a lot of times, those genes that are mutated, because of the redundancy of the genetic code, aren't really going to even show up. But most of the time, if they do have an impact on that species, it's usually negative, because really a species is a highly regulated set of genes that need to do specific things. And if you start to tweak with that, they can have unintended consequences. In humans, it's typically that we see these genes become cancerous, and it can be very deleterious to that individual that has that mutation. But a few times, these mutations can be selectively advantageous. And so they can actually improve the fitness of something in this population. A lot of times in plants, we have what we call conserved genes. And these are genes that don't have a lot of variation to it. So if you think about it, one of the conserved genes that we see in plants are genes associated with the chloroplast. Because if you mess with that chloroplast, if something mutates, you could be in a lot of trouble. You might not be able to get your food anymore. We also see plants do something that's really kind of unique within the tree of life. And plants are really masters at this process called polyploidy, which is essentially they're duplicating their genes. So instead of having just this kind of diploid 2N state, they might have a mutation that create a 4N or a 3N state. And this has all sorts of consequences, not only for the phenotype, but also for their reproductive system. Because if you're doing meiosis and you come up with different chromosome counts, you can't combine them with another organism that has different chromosome counts. You end up with kind of junk DNA that usually produces inviable offspring. So these mutations could rapidly lead to speciation or other evolutionary changes within this population. The next big way we see allele frequencies changing in populations is through gene flow. And this is just simply defined as movement of genes from one population to the next. Again, plants are able to do this through things like pollen, their seeds, or spores can move and blow those or get transported by organisms into far off lands where they grow and start to breed with individuals close to them. Plants also, because of a lot of their abilities, especially at the cellular level, are able to spread through fragments a lot of times. In fact, aquatic plants are very successful at getting just a chunk of them torn off and then transported large distances by an aquatic bird or something like that. But gene flow is really particularly important when it comes to what we call ecotypes. So in our population here, I kind of put down some edaphic soil conditions. And we'll say that this is a serpentine soil. Serpentine soils are really common in California, like you can see over here on this map, where there are areas of the ancient seabeds that have been lifted up really fast, and they're full of very highly toxic metals. And these naturally occurring toxic metals are hard to live in, especially for plants, because they're almost overloaded with things like copper and magnesium that can be toxic to the normal function of these plants. But we do see in these serpentine conditions, a lot of plants are kind of locally adapted to them. And so what happens is if a plant kind of becomes adapted to these edaphic conditions, that their gene flow might be disrupted, because the genes here aren't going to work that well down here in our serpentine soils. And so it might be a way to kind of start stopping gene flow as these plants that are adapted to serpentine are kind of figuring out ways to make sure they're not getting bad pollen that's going to put their offspring at a bad disadvantage here. One of the most confusing routes, I think, for a lot of students to kind of get their heads around is genetic drift. And this is a really important process, especially in groups of organisms that have relatively small populations. And genetic drift can be thought of the drunkard's walk of evolution. It's kind of random changes that are able to, what we say, fix genes. So in this case of our population, we can say we have populations up further north and further south. And let's say a drought hits. But for whatever reason, these plants here are able to survive the drought, likely because there was some water that was there or available to them. And so what happened to this population has now become fixed for being yellow plants. Not because that these yellow flowers have any particular adaptation or advantage over the orange one, it's just because by random chance, they didn't get killed by this selection. And so now all of a sudden, this population is what we call fixed. So the main key here with drift is this random process that has occurred. And probably one of the most intriguing and most kind of studied mode of evolution that we have, and really the one that made Darwin so famous, was evolution through natural selection. And in this, it's pretty simple strategy, right? The particular allele is favored. And because of this allele has some kind of advantage to it, because of its traits, that the fitness is going to be altered. So in case of this population, let's say that they're pollinated by insects. And we have these kind of random yellow mutations. But in this case, the bees that are pollinating them don't like the yellow flowers. They want to prefer to go after the orange flowers. Because one thing we see in a lot of these insects is they like to go after the flower that's most abundant, because they know that from experience that's going to get them somewhere. And so what we see is that these orange plants are going to have much, much higher fitness because they're actually getting pollinated, whereas these yellow ones aren't going to get as pollinated as much. And if they need another plant to help them get genes to reproduce, they're going to be in a lot of trouble because they don't have those pollinators there. So this is all great to kind of think about in a theoretical sense, but a lot of these processes, they act on real populations at real genetic levels. And also, it's not only just one of these that's kind of pushing a population to change or diverge. And usually they work in tandem quite a bit. And so the best example of this I like to give is from a group of organisms that's near and dear to my heart, the monkey flowers. And within the monkey flowers, we kind of adopted them as a really kind of neat way to look at evolution, because they have a lot of very simple genes that work with very simple rules relative to other plants. And so usually when we see evolution start to shape and change these different clades here, what's going to happen is a mutation is going to occur, and then selection is going to start acting on them. But even with a mutation and selection kind of acting on them, you don't generally see speciation because all those things in the population would probably just get pushed one way. But really what gets intriguing is when we add gene flow or drift to it. Because then we can start dividing up that genetic partition and we start to see populations diverging from each other that give us this result of these kind of different traits that we see here. So the famous example of this is Mimulus cardinalis and Mimulus lewisii, two monkey flower species we have here in Utah. And what's really nice about monkey flowers is we know that the color of these flowers are controlled by just one single gene. So one mutation can radically alter these flower colors. And another thing that we've noticed is that birds generally see really well in the red spectra. And so they usually like to pollinate and go after nectar rewards from red flowers. Whereas insects see a much different spectra, and they also are much more attentive to smells. So we see a lot of insects going after smelly flowers or flowers that have kind of pinkish or lighter hues to them. And so what we see in the wild is that cardinalis is a hummingbird pollinated flower and lewisii is a bee pollinated flower. However, within this group, what we see is that mutations have arisen quite a bit. In fact, in Mimulus lewisii, we often see these flowers get a tweak for some reason, usually a mistake in meiosis that occurs, and these pink flowers become these red flowers there. And so when we have a population with some red flowers coming on, we have a bee here. It's going to be going along, selecting for something, and it's going to come up to that red flower, get really confused, not want to land on it, and just keep on selecting for those pink ones. Normally, this would result in these red flowers just having a selective disadvantage and not being able to reproduce. But what we do see is that these red flowers become immediately favorable to hummingbirds, and they really preferentially go after these kind of mutant flowers. And so what happens is you have different organisms selecting for different phenotypes. And generally what you start to get because of this, because the hummingbirds aren't going to the pink flower and the bumblebees aren't going to the red flower, you get what we call reproductive isolation. So these two plants, even though they came from similar ancestors, stop sharing genes and they start to diverge, and eventually we consider them different species. And this result of kind of subtle changes here and there has resulted in this massive speciation within the monkey flower clade. For example, cardinalis up here, this red flower, is more closely related to these lighter color plants and not the rest of the red ones down here. Because one genetic mutation, probably right back down here in the tree, led to a divergence and kind of this evolution of a hummingbird pollinated flower. That really isn't as closely related to the other hummingbird pollinated flowers as the other. And this continuing selection pressure really has made the monkey flowers a very unique and diverse set of plants. So just to quickly review this, evolution works through acting on genetic variation. And that genetic variation is very obvious throughout the history of plants. And generally, it comes arise by mutations and tweaked by flow, drift, and natural selection.